I think it started now.
So, hello, this is Arunangshu and this is Manjul. Hi. Uh, Happy New Year to everybody. So, as Sir said that we work with Sir. This is the BRL laboratory and we are here involved in recombinant protein expression, purification and whole kind of metabolic engineering stuff in order to make our recombinant host more fitter to make them more fit to handle more stress and make more protein and survive better in a bioreactor. So these are the kind of stuff we do. And as Sir has said, introduced everything. So I hope, but we still feel that it is very essential that we always walk through the basics before working in the lab. And so we will walk you through some basics, and we have tried to shoot some videos which we will be sharing with you. I was trying to put all these things in the presentation, but there was some lack of time, so I will be just giving you one by one. So again, this is the introductory slide. So this is the first thing that we often forget in the lab. I hope everybody can see my slides, right?
Yes, they are visible.
Yeah, okay. So I hope these are the very basics you all know, and these are the very baby steps of getting into a lab first. Many of us sometimes forget about this, but this needs to be told again and again, I think. And next is that this is the introductory video from Dr. Manjul. So I will start the video.
Excuse me, sir, we can't hear the audio from the computer.
You can't hear the audio?
No, sir. So it might be an issue with the sharing. You have to, I think, share with the audio also shared.
Include computer sound. Okay, I think this is.
Hi all, this is Dr. Manjul Tripathi.
Can you hear now?
Yes, sir.
Hi all, this is Dr. Manjul Tripathi from IIT. Today I am going to tell you what to wear and how to enter the lab before beginning any experiment. You should always wear a lab coat, safety glasses, mask, probably the reason you all know, and shoes.
So moving on next, I hope you all know about biological safety cabinets. Anybody has any idea what are the types of biological safety cabinets? And hello, can you hear me?
Yes, sir.
So any idea? Have you ever used a biological safety cabinet?
No, sir.
In a very general language, it is called hood. So we call it the hood, the biological hood. Okay, fine.
We have used a few more.
Okay, so okay, the fume hood is used in basically in chemistry. So you know that toxic fumes and all those things which are generated during a chemical reaction has to be exhumed and ventilated. So that's typically why you use a fume hood. And in biology, we use the biological safety cabinets, and these are meant to protect us from the organisms which we work on, from the spores and all kind of spillage. And it is also meant to keep our cultures and other stuffs sterilized, because the most important problem in bioscience is the contamination, because there is so much possibility of cross contamination and different organisms growing together. So we want to keep everything sterile as far as possible in order to give meaningful, we have to obtain meaningful results from our experiment. So the first important introduction is the biological safety cabinet. So biological safety cabinets protects you and keeps your culture sterile. But there are different classes of biological safety cabinets, and these has been recommended by World Health Organization for the procedure for performing culture and procedures inside the lab. So since we are working with a non-pathogenic variant of an E. coli, and these cells actually lack the pili, which are used by bacteria to adhere.
Sir,
Hello.
sir,
Hmm.
sir, my name is Devpri. I'm the convener for the 2019 batch.
Okay.
Sir, we also have to do this BBL433 practical. So, sir, but we don't have access to this channel. So, how are we supposed to join this meeting?
Uh,
Like, I'm currently using one of my seniors' laptop to talk to you.
okay. I think I will pass it to KJM. Sir, can you make them join the meeting? They wanted to join. Just give me a second. I will just try to give it. Can anyone connect to sir? I'm not able to connect him. He in the meeting? I don't think he is in the meeting. Or is he? Okay. Can you guys listen? Hear me? Hello.
Yes, sir.
Okay, so actually, I am unable to figure out how to make you other people join because I am doing this for the first time. Matlab, for this course. So what I can do right now is that you all continue with us, and I can have another session for the people who are not able to join. That's the best I can do until and unless I can figure it out. I can communicate with Professor Mukherjee if that is fine. So should we move ahead? So okay, so I am going ahead with the presentation, and And I will put the presentations in the this channel folder also so that everybody can. So obviously now let's go through it. Okay, so we were in the biological safety cabinets. So these are actually designed in a specific purpose in order to protect you and maintain the sterile environment so that your cultures don't get cross contaminated and you don't like have at the end of the day have a mixed culture or that so so these are the some of the specifications. These are very much available online. You can go and you can look at them. And I would just like to show the types of why how the biology safety cabinet actually protects you from the organisms or from the biological hazards and keep your culture sterile. So basically what we have is that there is a directional layer flow through which goes around the food, and there is a particular filter called the HEPA filter. So the HEPA filter is called the high particulate filter, which prevents all kinds of dust and spores. They all get filtered, and they are really expensive. And so the HEPA filter is actually attached to the hood, and the air actually flows being filtered through the HEPA filter. So it filters out all the spores and cells and whichever whatever there in the air, I think including viruses also. And it also prevents like this PM particulate matters in the air. And so what happens is that this air flows in a particular direction, and it creates a shield of thin air flow between you and the internal environment of the hood, and only your hands go in, and you work with the hands, and does anything that is being exhumed from the hood then gets with the air and gets filtered with the HEPA filter, and then it is being exhausted out in the atmosphere. So this is the class 2, and these are the types of the safety cabinets. So we are here working with, as I told, non-pathogenic E. coli. So we are working with this class 2 type A2 type of biological safety cabinets. And for our the hood I am going to display here through the video has the same structure as this one, and the air goes out from the bottom of the platform on which we work. So there are also class 2 type B, and for pathogenic organisms there are class 3 and class 4. Class 4 provides the highest level of safety, in which you have the gloves already attached to the cabinet, and you have to put your hands inside the gloves and work with that. And they have in situ sterilization systems for culturing like highly pathogenic viruses and bacteria and other organisms. So okay, so I will back. I will be back. I will going back to Dr. Manjul. So this is the video we showed for you regarding the biological safety cabinet. So I'm very sorry because lack of time we were not able to put it in the presentation, but I hope you can see it, and you can look at how we work.
Type 2, the key components are airflow, normal light, and UV light. Before beginning any experiment, we switch on the UV light at least for 10 to 15 minutes. After that, we switch it off, open the airflow, normal light, the hood is ready for use. After that, wear the gloves, wipe the surface with 70% alcohol. Switch on the lamp. Take the thing for inoculation and discard. This is very important. explained. We have done the inoculation. This is a biosafety cabinet which is used to culture bacteria and other non-pathogenic organisms. Before switching the wood off, we should wipe the surface, arrange the things at the side, light off, airflow off, switch on the UV for 5 minutes.
So this was the short video again. So if you guys have any questions regarding what we have shown you, you can ask us now. Hello. Anyone is here in the meeting?
Yes, sir. It's clear to us.
Okay. So this was the first demonstration of how we work in a biological safety hoods. So another thing that she was when she was demonstrating, she was giving she was talking, so that you need not to do, because you shouldn't talk inside the hood. As it was a demo, we had to say what we were doing. I hope it is clear up to now. So now again, the this was how we cultured the organisms, and this was actually the simplest work we do inside a biological safety cabinets. Where when we will be doing metabolic engineering or like that of stuffs, you have to do electroporation. You may have to do spot assays. You have to work with we work with lambda phages. These all works has to be done inside the hood, and you have to get proficient using that small space in order to do so much work. So you have to manage yourself and become proficient using that small space to do a hell lot kind of things. And even you have to prepare com cells and all those things. So there will be a lot of stuff inside that, and you have to manage the space and move your hands, and again keep your head out, and that's the way to do it. Okay, nice. And now we will say something about the basic chemicals that are usually required all the time, and they should be kept handy beside you whenever you start work in the lab. And we have also prepared a video for that. I will show you that thing now.
There are certain must-have chemicals which are to be present always in the lab. One of those is 70% ethanol. It is all-purpose disinfectant which is used to clean the working place, your hands, pipettes, etc. The second most important is 0.5% hydrochloride solution. Whenever we discard any of the cultures, we have to add 0.5% hydrochloride solution to the culture and keep it for 10 minutes before discarding it to the sink.
So this was about the chemicals that comes handy and should be kept with you. So as simple questions people often ask, why 70% ethanol? Yes, obviously it saves some of your ethanol, but it is 70% because 100% ethanol is not permeable to the cells, and 70% ethanols are partially permeable to cells, so they can do damage both from outside and inside. So it saves your ethanol, and also it is more effective than 100% ethanol. That is number one. Number two, hypochlorite solution comes at 4 to 5% concentration. We call it the bleach solution. And they generate nascent chlorine, and nascent chlorine kills bacteria. And any culture before, whether it is a fungus or it is a bacteria, you have to add the hypochlorite solution and you have to discard them. That's how you should do science responsibly without harming the environment and without causing harm to the others. Obviously, there are a whole lot of protocols that need to be followed for different organisms and different kind of biological works you do in your lab. There are discard protocols, and they have been determined by your local authorities using, including municipal authorities, the Central Pollution Control Board, and all those things. There are norms how you discard the biological material. There are also designated color coded bags, if you know, they are used for different disposable or disposing different kind of biological stuff. Like for human anatomical and surgical stuff, you use a yellow bag, which should be puncture proof. We generally use a red bag for this kind of bacterial or all those cultures. And there are the other color coded things. There are white bags, there are black bags according to this, but mostly in our lab, where we mostly do E. coli and fungal cultures which are non-pathogenic, we use the red bag. And we using the proper color coded bags actually makes the persons who collect or the agencies who are involved in collecting your disposed items know that how to discard and dispose those things properly according to the local regulations and the environmental regulations. So I hope it is clear up to this. So any questions regarding this till now? Okay, so we will move on to the next the important thing, that is, as I told, that there are when we do experiments, we have to be there is no actually, you all know because you have studied mathematics, there is actually no definite numbers. So there is always a variation. If we measure the same item again and again, we will have different readings all the time. So really doing experiment is for us is not like finding the exact value of something, but what we try to achieve is precision. So in order to achieve precision, two terms has been used by sir, and you all know about it, I hope. There's the replicates, and we divide it in two segments. One is the biological replicate, and another is the technical replicate. So the biological replicate, so what is the difference between a biological replicate and a technical replicate, and why we use them? So basically, today I am going to show you this with a simple and another simple video of taking optical density in a spectrophotometer, in a simple spectrophotometer. So the technical replicates actually tell you what is the variation in the instrument, what is the variation in the basic level of noises in the electronics and all those things. So it takes care of the instrumental variations or instrumental noises. And the biological replicates actually takes care of the process what we are doing. Like when we are taking an OD, so what we are doing is that we are trying to measure how much cells are there in a culture, how much growth has been attained with a specific amount of time. So when we take a biological replicate, what we do is that we perform the same operation a number of times in order to see that the operations we are doing are actually precise. Like you are making a dilution of your culture. You can you add 100 microliter of your culture to 900 ml of your 900 microliter of a diluent. So you dilute your culture approximately 10 times. So what happens is that now how do you know that your pipettes are working fine? How do you know that there is a precision when you are making this dilution? So for taking care of all those things, we make use the biological replicate. So again, I will demonstrate you with a video that we have shot for you, and I hope you can watch it.
This is spectrophotometer. It is used to take the optical density, that is OD of the culture. By OD, we measure the number of cells present in per ml of the culture. Roughly 1 OD means 10 to the power 8 E. coli cells. Okay. Before moving to the use of the instrument, I would like to tell you about two terms: technical replicate and biological replicate. By technical replicate, we mean that we are taking readings at least thrice from the same instrument at the same settings, so as to know the variation in the measurement of our instrument. And by biological replicate, we mean that we have made two dilutions, same dilutions from the same culture. We make dilutions and measure in cuvette. This is cuvette, and we always handle it from the opaque side. This is the opaque side, and this is translucent. We take the dilution in the cuvette, and here observe the arrow which is indicating the path of the light, and we align the cuvette according to the path of the light, like this. We close it and measure our OD. Here we set our instrument because we are taking absorbance at the single wavelength, and our wavelength is at OD 600. First, we set the blank. The blank is the media or the water in which the culture is suspended. Our dilution has been made. We set the blank as 0, and after that, we take the sample, align it again, and read our sample for the OD.
So, okay. So this was the demonstration of how we take the optical density of E. coli cultures in order to study growth curves, and in order to also we need to we do assays like spot assays in order to see the fitness. Suppose you make a modifications inside the cell. Now you don't know how the cell whether that modification causing this causing a compromise in the cellular fitness and all those things. So you need to take ODs of the culture frequently. And and apart from that, regarding the biochemistry work or regarding the protein work, what we need to take is that when we purify plasmids, when we purify genomic DNAs, or when we purify PCR products, we need to take the optical densities at 280 nanometers to see the DNA concentration. We take the 260 by 280 ratios in order to see whether there is a protein contamination in the DNA we have purified or not. We take the 260 by 230 ratio in order to see 280 by 230 ratio in order to see whether there are reagent contamination, like there are ethanol contamination, phenol contamination, or anything like that. So those readings really matters very much, because when you are submitting the sample either for the next downstream process or for sequencing or for any of this, you need to be absolutely sure that these contaminations are not there. Why else you will not have a proper read when you are sequencing the samples, or you can end up with poor response of the downstream processes. So the spectrometer is one of the most handy instrument and the most useful instrument in the lab, and it is useful for all different kinds of purposes. These were very basics, and you can also use for protein estimation. We do Bradford estimation, we do Lowry method, and now there is Pierce BCA kits available for protein estimation. And we use the spectrophotometer to find out the intensity of the developed color with the standard. The BSA, the bovine serum albumin, is the standard. So it is a very handy instrument, and everybody should be well acquainted with the functioning of a simple spectrophotometer. And So these are the some of the basics. And apart from this, every laboratory, perhaps the beginning of a laboratory starts with two basic instruments. One is the balance. The balance is the balance of the laboratory, because without a proper balance, a laboratory cannot function. And the second such instrument is a pH meter. These two instruments are the heart of the laboratory, because any experiments that need to be done starts with a balance and a pH meter. Working on biological stuffs, whether it is a biological assay, whether it is a protein purification assay, or whether it is an enzyme assay, is all about buffers. Is all about buffers all the time, and you need to prepare buffers as frequently you are designing your experiments. And all buffers need to be checked. Their pH has to be adjusted. Their pH has to be checked again and again during the time they are preserved, and they are used again and again, so that there is no variation. So these two instruments are really important. And I will also tell that these instruments need to be calibrated as often as you could. Most of the times what happens is that we are not able to take care of the balance, but the balance, there are calibrated mass, like there are calibrated mass feature available, and you need to calibrate your balance as frequently as you can with those calibrated mass. They comes in a box. And there are buffers like pH 4, pH 7, pH 10. These are the standard buffers. You need to calibrate your pH meter as frequently. The way I do it is that I every morning, after work, I just close the pH meter and go. And in the morning when I come, I just switch on the pH meter and first calibrate it with the three buffers. Once you calibrate it and don't switch off the pH meter, you can take the reading for the whole day. But once you switch it off, you have to recalibrate it again. So these are some small things. And I hope this was sufficient for today, and this was actually how much we could have also managed for you. So any questions, we will be happy to take and try to answer. Okay, fine. So there is no questions, I hope. So I hope you enjoyed the class. Yeah, you can also, if you have any questions, you can leave questions for us, and you can mail them directly to Professor Mukherjee, and either he will answer or we will try to get back to you regarding any questions. So once again, have a nice day, and thank you so much for being here.